First of a big thank you for the invitation by the organizers to talk about the other Oncode indeed. I still see you said sister, so let's keep it to sister, not brother. We are the older sister, but it's good to see by the audience that the younger sister has grown up and is becoming quite mature. So it's really nice to present what Oncode Institute has been doing for the last 7-8 years, and to try and build from innovation, innovative science to impact, and the toolbox that we created to actually get to that point. Now, to just give you a quick overview of what Oncode Institute is, I think it's a unique model that looks quite similar to the accelerator model. It's a virtual institute. As you can see, we share not only the name, but we share a couple of partner institutes. We share quite a number of researchers that are part of Oncode Institute, as well as Oncode Accelerator. We share the same tagline, trying to make impact by outsmarting cancer. And now for those of you that don't know, the balls and lines that should be moving are actually our DNA that is continuously changing in cancer. Most of the people that actually work in Oncode Institute or Oncode Accelerator don't know this, but this is the idea of this bubbly logo. So as said, Oncode Institute, 13 partner institutes, all listed there. I don't think you can read them, but they are throughout the Netherlands, and we actually use that infrastructure to build our science. 62 top research teams have been selected in the Netherlands, and they represent around 750 to close to 1,000 researchers that are part of Oncode Institute. Now, in contrast to Accelerator, we're financed by the Dutch Cancer Society and directly by 3 ministries. So we are not a growth fund, but we are really dependent on a different type of funding. The majority of our funding actually comes from the Dutch Cancer Society. And the way we spend that money is actually by investing in those research teams. So we really try to build on a very strong Dutch network of scientists that are already for years performing excellent research. And we actually allow them to do even better by providing funding that they can spend as they see fit. So it's really base funding, as we call it, in trust. They know what to do, and they know what to do best. Then we stimulate them to collaborate, not only within the institute, but obviously also outside. But we see that especially when disciplines meet, things happen. And then I think the key trick to the impact part is that we have an excellent valorization team that actually goes into these laboratories, talks to the scientists, and proactively brings in knowledge from these research teams to drive it towards valorization. And I've seen some of these business developers in the audience, so if you're interested in how they work, talk to them. Does it work? Well, I think this is a long list of a lot of numbers, but I think it's quite revealing. A couple that I really like a lot is the lower one there, is that if you look at the science we do within Oncode Institute, we actually are number 3 worldwide, meaning that we outperform in terms of impacts on our publications. Institutes like Harvard, UCLA, MD Anderson, we actually score better in terms of our science. I think this is really showing that the Netherlands is an enormous stronghold of science in oncology. I think one in the center that I would like to show, because that's going to be the rest of the presentation, is the fact that we have a bridge fund that we use to actually start up new companies, and we have some really good news today. That the number that is listed there that we bring in, so we invested around 5 million euros. Right now we brought in 80 million euros from private investment into those companies. So I think this is really an impressive number that allows us to really push our work forward. I'll just skip this in terms of time. So where do we act in the actual process of innovation? Well, the majority of our funds actually goes into the first part. So in the purple part, the base funding is where the majority of our funds are actually invested in the science of our capital, our researchers. But we have also over the years developed quite a number of tools to really push that innovation towards impact. The first one is a very, well, as we say, a plotter in Dutch, it's the IP fund. We actually spend quite a bit of money on a yearly basis on our IP portfolio. That amounts right now to 400K a year. So that is really serious money that you actually need to protect all that new insight that we generate. Then we have a second one that actually allows us to really pinpoint new developments and bring them one step further to making impact. So a lot of those innovative findings that come from academia, and as you alluded to, I'm an academic myself, don't really meet any of the standards that you need for further development. So we really need to optimize the innovation, improve on a drug composition, or improve on a platform to really allow these to move forward. And this is done by the Tech Development Fund. Then we have, as I said, the Oncode Bridge Fund that really uses a relatively small fund, but it uses the millions to invest in startups. We have started up right now 12 small companies, of which I'll show a couple in a second, that are bringing quite a number of funds. It says 68, but as of today it is now 80 million, as we had an enormous Series A investment in LIGO Bio announced today. And last but not least, so these are all the companies that we've started. I'll tell you something about some. Last but not least, we also have a clinical proof of concept fund, where we spent around $1.5 million a year on average over the years to really kickstart clinical trials. Most of these are phase 1, 2, but we actually have a couple of trials also that are pushing forward towards 2, 3 stage. And this really allows us to bring innovations into a clinical perspective. Now, the first one I would like to allude to is the one from Jeroen de Ridder, who really made optimal use of all the complete toolbox that we offer at Oncode Institute. Basically, the concept came from the Máxima Center, where they were struggling with operations of pediatric brain cancers, which are very heterogeneous. And it's unclear for the surgeon when they do the operation whether it has to be an aggressive or a passive operation. And normally what happens is that pathology checks the operation material afterwards, and a reoperation may or may not be necessary. Jeroen said, I think we can solve this issue by just sequencing on the fly while the operation is going the material from the patient and determine what the actual subtype is. That must be something that we can do in, I guess, around 2 hours' time. Surgeon said, well, that's perfect because we spend normally 8 hours in the operation theater, so if you manage to do this, you can actually inform us on the fly. This worked out perfectly, AI-supported toolbox, and they now do this in around 1 hour and 20 minutes, I think is what their record is, to actually determine the actual subtype of the cancer and informing the surgeon. They use the Oncode IP fund, obviously, to protect this finding, the Tech Development Fund to optimize the technology. A license was formed, a spin-off is created, and it's now being used in clinical practice. So this is one of the first examples that really brought it from the lab to complete innovation to patients. Obviously, it's a very limited number of patients, but I think it's something we're really proud of. Second, and I'll speed up because of time, is from Madelon Maurice. Basically, Madelon has been working on E3 ligases, driving proteins for degradation. And what she decided to do is to use the E3 ligases on the surface of cells and bring those together to targets that she wants to degrade. And the example you see here on the left showing that it actually works, the color is the ligand that she's trying to get rid of, is in this case PD-L1 for immunotherapy, obviously the one that we want to get rid of, and it works out perfectly. This technology is now strongly under development. She used the base funding to optimize it. She used all our portfolio to really get the technology into a startup. And as said, today they received a Series A investment of 11.5 million to drive this a couple of steps further. I think this is another one that we are really happy with. Last but not least, quickly is Sjoerd van den Burg, very well known to you all, as he's part of the Accelerator program as well. And basically, and this is a development that's been going on for well over 20 years. Actually, the first author here was sitting next to me when I was still working in Leiden University. And he was already working at it at the time. It's basically the concept is that tumor cells escape from immune surveillance by shutting down their peptide processing, by shutting down the TAP proteins. And by doing so, they actually expose a complete novel set of neoantigens, which are called types. And these types are actually beautiful potential targets for immunotherapy. And this again was optimized initially with base funding. The IP on this type peptide was filed. The CPOC was used to create the first clinical proof of activity. And also here, I'm happy to announce that this is number 12 of the companies that we started from Oncode Institute, called Revialis Biotherapeutics. And I think this will also be one that will quickly move into the next phase. Now, it is already moving into this phase. It's one of the demonstrator projects that was awarded. And I think they're developing this technology together is really what makes our impact work. So I think this is where I would like to leave it. Collaboration in this perspective is key. We have an enormous powerhouse in the Netherlands, which doesn't really match the size of the Netherlands, but it's really the strength of oncology research is amazing. Let's really build this to bring impact to patients. This is a signing exactly 1 year and 1 day ago. Where's Mark? Yeah, there you are. Where we signed an agreement with CRH to really collaborate and work together to bring this all further to clinical impact. And I'd like to leave it there, and I thank you for your attention.